how do cells communicate? How does a fat cell in our body know when to store energy or when to release it to nourish the rest of our body? How does a muscle cell or a skin cell know when to repair themselves? And how does a cell know that it has been infected by a pathogen or has gone rogue and precancerous? These are some of the central questions that have motivated me throughout my research career. My name is Keren Hilgendorf, and I'm an assistant professor in the Department of Biochemistry. I was born in Israel, but grew up in Germany before moving to Texas as a teenager. And it was there, as an undergraduate at the University of Texas, that I first started asking these questions, and I fell in love with research. I decided to move across the country to pursue my graduate work at MIT with Dr. Jackie Lees. During my PhD, I discovered a novel function for a commonly deregulated tumor suppressor, the retinoblastoma protein, in triggering cell death. It was also during my PhD that I first became fascinated by the complexity that is intracellular signaling, how information that is received at the surface of a cell is transmitted to other parts within that cell to elicit the appropriate response. I also became interested in intercellular signaling, how information is transferred from one cell to another. And I decided to once again move across the country, this time to Stanford University, to pursue my postdoctoral training with Dr. Peter Jackson. Dr. Jackson is a world-renowned leader in an antenna-like signaling organelle called the primary cilium. And you will hear about the primary cilium more in just a few minutes. But during my postdoc, I discovered, that the ciliary antenna is required to instruct our stem cells to make more muscle cells and more fat cells. 3.5 years ago, I accepted my current position here at the University of Utah, studying how obesity changes the signaling network within our body and how this leads to diseases such as diabetes and cancers. But why study obesity? Well, we live in an obesity epidemic. 50% of adults globally and 70% of adults in the US are with obesity or overweight. According to the World Obesity Federation, the global economic cost of obesity is predicted to surpass $4 trillion by 2035. That's roughly 3% of the global GDP. And that doesn't take into account the immense personal cost of obesity and its many comorbidities, which can range from depression to cardiovascular disease to type 2 diabetes and cancers. In my lab, we are investigating how obesity leads to these diseases. But what is obesity? Obesity is defined as an expansion of our fat tissue due to caloric imbalance between utilization and consumption. But where and how we store those calories actually matters. Our body can store these excess calories either in existing fat cells, making them larger, or it can make more fat cells to store the excess calories. However, having excessively large fat cells is linked to the infiltration of immune cells, which are these little red and blue cells I'm showing, as well as stiffening of the fat tissue and inflammation within the tissue, all of which result in loss of fat tissue function and ultimately lead to diabetes. In contrast, having the ability to make more fat cells to store excess calories actually protects us from diabetes. But what determines how our body will store excess calories? Why is it that some individuals with obesity become diabetic while others are metabolically completely healthy? Well, to make more fat cells, our body has to activate fat stem cells, which sit along blood vessels within our fat tissue. But what are the signals that activate a fat stem cell? And why do these fat stem cells become non-responsive to those signals in settings linked to diabetes? As I introduced earlier, during my postdoc, I discovered that these fat stem cells all express this specialized antenna-like signaling organelle called the primary cilium. And this primary cilium is required to sense the signals that activate the fat stem cell to make more fat cells and to expand our fat in a healthy manner. We have since discovered that this primary cilium actually contains many receptor proteins that can sense a variety of signals in the environment. And in my lab, we're investigating how these signals are received, who sends the signals, and how the signal is transmitted to the body of the fat stem cell to regulate the generation of more fat cells. We've also recently discovered that the ciliary antenna is actually lost in settings linked with diabetes. Here are some microscopy images of fat stem cells that we isolated from lean donors and mice. In blue is the body of the fat stem cell. The red dot marks the base of the primary cilium, and then the green line is the actual ciliary antenna, the structure that is so important to sense the signals to then make more fat cells. So fat stem cells isolated from lean fat tissue is uniformly ciliated. However, when we isolate these fat stem cells from donors or mice with obesity, this picture looks completely different. Now fat stem cells no longer have the primary cilium, and they're unable to sense the signals that tell them to make more fat cells. To pursue those exciting lines of investigation, I was recently awarded a prestigious Early Career Investigator Award by the Pew Charitable Trusts, as well as funding from the National Institutes of Health. But diabetes is really only one of many comorbidities of obesity. Obesity also predisposes to 13 different types of cancer. In my lab, we're studying breast cancer, where studies have shown that each 5-unit increase in body mass index, also known as BMI, is linked to a 12% increase in the incidence of developing postmenopausal breast cancer. But why is that? What is it about obesity that accelerates breast cancer cell growth and metastasis? Well, breast cancer cells are really surrounded by the expanded fat tissue in patients with obesity. And in my lab, we're investigating how breast cancer cells communicate and change the surrounding fat tissue, and in turn, how the surrounding fat tissue communicates and accelerates breast cancer cell growth and metastasis. We've identified several targetable secreted factors that mediate this communication, and we hope to one day develop treatments against obesity-accelerated breast cancer. And to study these lines of investigation, I was awarded 2 Early Career Investigator Awards, one from the Victory Over Cancer Foundation and one from Five for the Fight. But to get back to the original question I posed that motivates my research, how do cells communicate? My lab is using a powerful combination of cutting-edge techniques on cell line models, mouse models, and on human samples to try and understand how obesity changes who in fat tissue sends signals and how these signals are received and interpreted. But my personal journey to answer these questions has not been a lonely one. With the support of my family and the help of my lab, my current and past mentors, as well as our collaborators and science friends from across the country and the globe, we hope to one day understand how obesity causes disease.